Good morning, friends. Good morning, guys. So let's start. So very good morning once again, and welcome to Unacademy. This is Abhijit, and today we are going to discuss about the light reaction. Okay, so it is a revision lecture, so we are going to end this lecture in our time. Okay, so we are not going to extend it. So we already discussed about the light reaction and the players which is involved in the absorption of light in the uh, process of a photosynthesis. Okay, so this part we already completed in last week, and today we are going to revise it because revision is very important, we know that. Okay, so this is a guys crack Maharashtra CET channel, and the name of batch is target top 100 ranks MST CET 2021. Okay, so the timing of math, physics, chemistry, biology is in front of you. So just join this channel, subscribe it, and click on a bell icon, so you will have the thousands of lecture available on this channel, and you can see the whatever topic you want to study. Okay, so before starting the lectures, guys, it is very necessary to know about me. This is Abhijit Madkar. I have more than 6 years of experience in the field of biology, and also crack a net CSIR from life science. Okay, so guys, is there any problem with audio? Is there any problem with audio? If yes, please drop in a comment box. If no, then also drop in a comment box. Is there any problem with audio? Very good morning, guys. Very good morning. Very good morning. So, guys, we are here on the India's largest platform. This is Unacademy. If you want to enjoy more about the education with Unacademy, so it is necessary to install this app in your mobile. This app is available on the App Store, and the link is given below in the description of this video. Okay, so this is Telegram app, and this app is for the daily update, guys. Here is the Telegram channel, Crack Maharashtra CET, and the link is in front of you. Okay, so this is the link for the channel, and just click on a link, and link is given in below in the description also. So just click on a link and join the channel. So the uh, after completion of a chapter, the MCQs and the notes are available on this channel, guys. So these are the few subscription from Unacademy. So the subscription for a 12 month is 14,850 rupees, and the 24-month is 21,600 rupees. So guys, this subscription will give you the different facilities like the MCQs for a practice. Then you will have the doubt solving session, special doubt solving session, and the you will have the chance to interact with your favorite educator. Okay, so the MCQs practice, after MCQs practice, you will have the analysis report, so the which question you make a correct and which type of a question you make incorrect. So guys, you will have some 100% direction about your study. So this app is I mean, these subscriptions are very necessary. When you take a subscription, so please use this Abhijit as a subscription code, so it will again reduce your fees. Okay, so it will give you the 10% discount on a fee. So use this code Abhijit at the time of a subscription. And guys, very important thing, when you take a subscription, please take it till the examination. Okay, so it make sure that it will not happen your subscription will end before the examination. Okay, so please take a subscription till the examination, so it will definitely help you in the last part of examination. So guys, let's see the light reaction. Okay, so we know that in plant a plant had a property to take a light and convert it into the chemical energy. Okay, so with the help of CO2 and the one more thing, that is the pi carbon atom compound. Okay, so this pi carbon atom compound and this carbon dioxide, so they comes together in the presence of a light, and they form the chemical energy, that is 6 carbon atom compound, which is then convert itself into the ATP, and this is the currency of a cell. Okay, so this currency is used for the different type of reaction which is happening inside of the body. So this is the complete pathway which is depending upon the light, and this pathway is called as the photosynthesis. Okay, so this is the actually pathway, we called it photosynthesis. So whatever I draw here, guys, so this is not a one reaction actually. This reaction is divided into the two. Okay, so one is called as a light reaction, and one is called as a dark reaction. So this dark reaction is nothing but your Kelvin cycle, and this light reaction is the electron transport chain. You can say the electron transport chain between PS1 and PS2. Okay, so this dark reaction is called as a dark because it is happening inside of a darkness. Am I correct? Okay, so guys, not like that. So most of the time we think like it is happening inside of a darkness, means only at a night, and that's why it is the dark reaction. Not it's not like that. So light reaction is a light reaction because it is depending upon the light. Okay, so light reaction is depend upon light, depends upon light. It is independent of light. So this Kelvin cycle or the dark reaction does not require any ray of light to proceed this reaction. Okay, but in a light reaction, if you see, if you stop the light or the source of a light, automatically reaction will be stop. Okay, so that's why we call it a light reaction and the dark reaction. So it doesn't mean that the one reaction is going on in the time of a day and the second reaction is going on in the time of a night. Okay, so it doesn't mean that. Please keep in mind that when you study about the light and the dark reaction. Okay, so it is actually the path to convert the light energy into the energy of a chemical, that is the ATP. Okay, so actually the energy which is inside of a light is coming from the sun. Okay, so how much energy is coming from the sun? If you say 1.5 into 10 raised to 22 kilojoule energy is falling on earth per day. Okay, so this calculation is per day. 1.5 into 10 raised to 22 kilojoule energy is falling on the earth per day. So 1% is absorbed by the photosynthetic organism and transformed into the chemical energy. Okay, so the only 1% energy is absorbed uh, by the photosynthetic organism. So this energy, absorption of energy, we know it is converted into the directly glucose. Okay, so the making of glucose is involved the fixation of a carbon dioxide, means adding of a carbon dioxide. So if you add a 6 carbon dioxide, so you will have one glucose like a C6H2 S12O6. Okay, so guys, if you see the only 1% light absorb and it is converted into the 10 raised to 11 ton of a carbon dioxide fixation. Okay, so globally means on earth per year the photosynthetic organism fix or the assimilate the 10 raised to 11 tons of a carbon dioxide in the presence of light, and they absorb only 1% of light till the number, if you see the fixed carbon dioxide number is 10 raised to 11 of tons. Okay, so this is the actually the efficiency, or you can say the the fixation rate. Okay, so how much light is absorbed. So guys, here is your reaction. So the 6 CO2 plus 6 H2O, they are coming together and they form the one molecule of a glucose and the 6 molecule of oxygen, which is released in environment in the form of a gas. Okay, so I am writing here G, and that is the oxygen which is available for breathing. Available for breathe. Okay, so this is the actually oxygen which is available for breathing, because guys, oxygen in environment, we have the largest source is photosynthesis. Okay, so the formation of sugar from carbon dioxide and water, it requires energy, and that energy is given by the light. Okay, so this is the energy actually the light gives you, gives the reaction of photosynthesis, which convert into the sugar. Okay, so let's say the harvesting of energy or the absorption of energy. So the absorption of energy required the player which catch the light, and the main player is your chlorophyll. So these molecules which is involved in absorption of energy, we call it the pigments. Okay, so the pigment system is always there. Pigments. Okay, so this pigment system is always there, and these pigments are involved in the absorption of light, and the first one is your chlorophyll. Okay, so the chlorophyll is a very important primary light harvesting complex which is present in inside of each and every plant. If you see the type of a chlorophyll, there are 7 type of a chlorophyll: chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D. We know that in a green algae, so A and B is present. In a brown algae, A and C is present. In a red algae, A and D is present. And the one more is chlorophyll E. And then last two are the bacterioviridin and bacteriochlorophyll. Okay, so these are the 7 chlorophylls. Type of chlorophylls are present in the different type of organisms. But if you see the chlorophyll A, okay, so this chlorophyll uh, A is present in each and every organism, and so it is called as ubiquitous. It is a compulsory present in each and every photosynthetic organism, guys, from bacteria to the higher plant. Okay, so from bacteria to the higher plant, in every single cell which is involved in the photosynthetic reaction, it will definitely have this chlorophyll A, and so it is a universal. So this type of chemicals or this type of things which is present in a biology, which is present in each and every organism, so we do not use the universal word for them. We use the ubiquitous word. Okay, so the chlorophyll A, your chlorophyll A is a ubiquitous, which is definitely present in organism if the organism is having the machinery for a photosynthesis. So chlorophyll A is compulsory, and that's why we know that the chlorophyll A is actually the reaction center. Okay, so which is involved in the conversion of a light. And what is reaction center, we are going to discuss about that. Okay, so the first thing, the chlorophyll, the first the player or the pigment which is involved in the absorption of light. So let's see what we know about the chlorophyll. It's a primary pigment responsible for capturing of light for the photosynthesis. Okay, so the single thing or the first player which is catching the light. Second, let's see, the chlorophyll molecule consists of a two part, porphyrin ring and the long chain of a phytol tail. Okay, so the porphyrin ring and the long chain of a phytol tail. So porphyrin ring means what, and the phytol tail means what, we are going to discuss about that, guys. Okay, so let's see the porphyrin ring. Here is the your porphyrin ring. It is a cyclic tetrapyrrole made up of the nitrogen containing pyrrole rings. Okay, so it is a nitrogen containing pyrrole ring. It is looks like a head above the, like this. It is looks like this head, and this having the phytol ring here. Okay, so this head is not like this. Actually, it is the ring compound, and we have the diagram of this ring compound. So the next point, incorporation of Mg ion in a 4 pyrrole ring complete the structure of chlorophyll. Okay, so here is the Mg present in a center of the perforyl ring. So the structure is now complete. So the Mg magnesium is present into the perforyl ring. So it is a part of a chloroplast. Okay, so the chlorophyll is having a magnesium at the perforyl ring. Please keep in mind that because it is not too easy to keep in a mind. So guys, if you see the perforyl ring is also present inside of the human body. Okay, so the RBC, which is having hemoglobin, so hemoglobin and the ferrous at the center of the ring. So that ring which is around the ferrous is also the porphyryl ring. Okay, so we are going to see that structure is also available in the slides. Okay, so the fifth point, a loss of Mg from the compound results in the pure. It's a non-green compound. So if you remove this Mg from the ring, so now this is no more longer the chlorophyll, and we called it the pure. Okay, so what is your chlorophyll? Chlorophyll is a pure plus Mg. So the pure is a non-green compound. Okay, it is not a green colored compound. So we are going to discuss the color of pure in next slide. Fourth ring of a compound is terrified with a 20 carbon atom alcohol phytol. Okay, so this phytol chain, what we talk about before, is a 20 carbon atom fatty acid like a chain having the alcohol. Okay, so the carbon alcohol, it is actually carbon alcohol chain. So we called it the phytol. So this is nothing but the phytol chain. This phytol chain is actually the insoluble in water. Okay, so if you see the membrane, we know in a membrane there is a lipid bilayer like this. Okay, so this lipid bilayer is actually the uh, lipid soluble and insoluble for water. Okay, so this lipid bilayer, if you want to put anything inside of membrane, so it is very necessary having the ability to dissolve in a lipid, and that's why this phytol ring dissolve in a lipid and having the head with a magnesium like this. Okay, so it will maintain the orientation of a chlorophyll, uh, and it will expose the chlorophyll to the light. Otherwise chlorophyll will not expose to the light. Okay, so the phytol is a lipid soluble, so help in holding up a chlorophyll inside the membrane with a proper orientation. Okay, so this is the first molecule which is involved in the absorption of a light. Let's see next one, the structure of a chlorophylls. Aromaticity makes it efficient absorber of a light. We know that the aromatic compounds having the very good capacity to absorb the light. Okay, so we already know that the aromatic compound having the very good capacity to absorb the light. So here is your structure of a chlorophyll. If you see guys, uh, this is a Mg2 plus molecule. Okay, so if you replace it Fe with a Fe, so this Fe is present inside of hemoglobin. Okay, and this is the Mg. So this is your phytol ring. Okay, sorry, porphyrin ring. This is your porphyrin ring, and this is your phytol structure. Okay, it's phytol chain and porphyrin ring. Okay, so this is porphyrin ring and the phytol chain here. Okay, so it is alcoholic and long chain carbon alcohol. So let's see the next one is phycobilins. Okay, these are the accessory pigments. Phycobilins are open porphyrin ring. If you break it into the third and the fourth ring, So it will gives like this compound, and this compound is nothing but the phycobilins. Okay, this pigment is covalently linked to the protein, so called chromoprotein. So this pigment is covered by a protein, so like this. uh, and because of that we called it the chromoproteins. Phyco comes from the algae, and it is not found in higher plants, but algae and the cyanobacteria contain this phycobilin. So please write it down somewhere, guys. It is only present in the algae and the cyanobacteria. Okay, so it is very similar like your porphyrin ring, but the third and fourth carbon chain is not having the good bonding between two, and that's why it breaks into the structure like this. Okay, so guys, if you have any question you can ask me here. लक्षात येते का रे मित्रांनो? तुमच्या पर्पोरिन रिंग सारखंच आहे, हीच पर्पोरिन रिंग आहे. फॉर एक्झाम्पल 1 2 सेकंड थर्ड आणि ही फोर्थ. ओके, इथे फोर्थ का काढले तर फोर्थ जो आहे तर तो काय करतो, या फायटॉल रिंग सोबत बॉन्ड बनवतो. ठीक आहे, मग जर तुम्ही थर्ड आणि फोर्थ मध्ये इथून तोडून टाकलं, तर तोडून टाकल्यानंतर हे स्ट्रक्चर काय होणार आहे, हे या बाजूला सरकेल आणि हे या बाजूला सरकेल, कारण हे ऑक्सिजन ढकलणार ते एकमेकाला. हे स्ट्रक्चर कसं दिसणार आहे, तर असं दिसतं. आणि जर तुम्ही थर्ड आणि फोर्थ कार्बन ॲटम मध्ये तोडून टाकला बॉन्ड, आणि स्ट्रक्चर जर जे तयार होईल तर त्याला तुम्ही म्हणायचं phycobilins. Okay, so the phycobilins is not present in a plants, it is only present inside of the algae and the cyanobacteria, and that's why it is called as a phyco. Okay, so the phyco comes from the algae, phyco word coming from the algae. Okay, so this is your the carotenoid. It is sometimes called as a color of autumn. So what is meant by color of autumn? Carotenoid is a family of orange and yellow pigments. So it is family of orange and yellow pigment. Please keep in mind these two colors. Okay, so because this is not a color of a single uh, pigment, it is two different pigments. Okay, found in a root of a carrot and tomato fruit, and also in a green leaf. So it is found in a root of a carrot, and that's why it is called as a carotenoid. So that is found in a carrot, you can say. The root means carrot is a modification of a root. बरोबर ना, का स्टेमचं modification आहे रे carrot. So carrot is a modification of a root, and it is found in a tomato fruit and the green leaf. Okay, in the fall of year, chlorophyll pigments are degraded, and more stable carotenoid pigments account for the brilliant orange and a red color. So it's a characteristics of autumn foliage. Okay, so आपण वर्षाच्या काही भागामध्ये पाहतो ना की प्लांट काय करतं, स्वतःची लीप टाकून देतात, नवीन लीप बनवायला सुरुवात करतं. ज्यावेळेस नवीन लीप बनायला सुरुवात होतात ना, त्यावेळेस हे जे आहे ना क्लोरोफिल पिगमेंट्स, तर ते लगेच तयार झालेले नसतात. त्यावेळेस ही कॅरोटेनॉइड्स तिथं असतात. त्यामुळे जे कोवळे लिप आहेत ना, यंग लिप, तर त्या यंग लिप मध्ये तुम्हाला जो रेडिश ब्राऊन कलर दिसतो, सो दॅट कलर इज actually the represented by these carotenoids. Okay, so because of that it is called as the color of autumns. Okay, so the color of autumns means it's a carotenoid. So carotenoids are water insoluble hydrocarbon chain found in a chloroplast membrane. Okay, it is present inside of a chloroplast membrane. Write it down somewhere. Beta carotene shows the orange. So carotenoid is again classified, I mean there is two member of a carotenoid present inside of the plant. Okay, so but sir, if carotenoid is a orange yellow pigment, then how present in a green leaf? Yeah, Mansi, actually what happened beta, so the amount always matters. Okay, amount always matters. So they are present there, but chlorophylls are also there. They absorb most of the part of the light, and that's why the other pigments they are also present there, but you can't see them because the concentration or the intensity of light which is coming from the one color maybe hide the second one if the more number of green rays are present, so your other rays are not going to be expressed in that light. Okay, so the carotenoid having two type, I mean two type of carotenoids are present into the uh, higher plants. So one is called as beta carotene, beta carotene, and the second one is called as xanthophyls. Okay, so guys, if you have any doubt, please revise my lecture that is on the name of lecture was the chlorophyll. No chlorophyll. The pigments in a photosynthesis. Okay, the name of a lecture was the pigments in a photosynthesis. It is available on the same channel. So please revise that lecture one more time. So that was a very detailed lecture about the chlorophylls and the different type of pigments which is involved in the catching of a light. Okay, so that is the very important lecture as per me. That is a very important lecture to understand the actually what happens inside of the photosynthesis. Okay, so this beta carotene shows orange red color. Okay, so let's see, it is showing the orange red color. Okay, and your xanthophyll shows a yellow color. Okay, so it is nothing but the carotenoid. Xanthophyll is nothing but the carotenoid, but only oxygen, extra oxygen is present. So in that lecture I was talking about, in that lecture I also add the diagrams. Okay, so and the structures inside of the slides, so you can see the difference between carotenoid and the xanthophylls. Only single oxygen is present, and because of that oxygen, the complete properties of the structure is going to be a change, and that's why we know we called it the carotenoids and the xanthopylls. These are the actually single thing. Carotenoids is a large group of a pigment which is having two categories, beta carotenoid and xanthopylls. Okay, so these are the very famous. That's why here if you see in a exam or sorry, if you see in any very good book, so you will have definitely the alpha carotene is also there, and the different type of pigments are also there. Okay, so it is very necessary to understand the actually how these pigments are looks like. If you say the structure of these pigments is the ring structure. Okay, they are made from the aromatic compounds, and that's why they shows the a property of absorption of light. Otherwise the absorption of light is not the easy process for the chemicals. Okay, so they all are made up of the ring compounds. Okay, so we have the pure chlorophylls and the pigments which is having the absorption in the different light. Okay, so here is for example 300 and here is 700. So we know this portion is comes under the blue light, this is coming under the green one, and this is coming under the red one. Okay, so guys, if you see your chlorophyll A is here with the faint green, and the chlorophyll B is with this one, the glowing green. Okay, so the chlorophyll B are showing absorption at blue light. Okay, so the more absorption at a blue light, and chlorophyll A showing absorption here at the red light. So the chlorophyll A having absorption at a red light. So please keep in mind that A having more absorption at a red light, and B having the more absorption at a blue light. Otherwise you can see the pure erythrin is a single component which is showing the absorption at the green light. Okay, so in a green zone. Otherwise the no one no any a pigment is present which is having the absorption at a green light, and because of that all the pigments are emitting the same color of a light, that is a green, and that's why the leaves are looks like a green. Okay, so the second thing, this red color pigment. Okay, so not red, brownish, so or orange. Okay, so this orange color is your carotenoids. Bacteriochlorophyll, so don't go in a bacteriochlorophyll, actually it is the uh, this is the bacteriochlorophyll. Okay, A, so it is showing the absorption at a IR zone also. Okay, so they are very intelligent, they can absorb the IR. Okay, sometimes they start photosynthesis in the light of a moon also. So they are very intelligent. We know that the bacteria is having the different type of a system in their body, and they can do anything in the nature. Okay, so phycocyanin is also there. It is showing the absorption uh, at the transition phase of the green and the red. Okay, so it is also present inside of a plant. So this is the actual absorption factor. You have to keep in mind the which part is responsible for the absorption of which part, and that's why the question is always there in examination. Chlorophyll B is absorbed more in blue light, green light, red light. So don't be confused. It's a blue light. Okay. So the reaction center and the absorption of light. So the reaction center means when the light is absorbed, so it is having the excitation of electron, and the electron is directly moving toward the reaction center. So what is meant by reaction center? So many chlorophylls are there inside of the this cell, but single reaction. Okay, so the many chlorophylls, or you can say the pigments, are present, but the reaction center is a one. The unit consists of several hundred light capturing chlorophylls plus a pair of special chlorophyll in the reaction center. So we know that the reaction center involved the chlorophyll A. Light is captured by one of the antenna. So antenna means the accessory pigment or the chlorophyll itself. Okay, uh, and routed from one to the other. So the absorbed light is also having the so let's see actually what happened inside of the for example, this is a chlorophyll, guys. So the light rays, when light rays comes inside of the chlorophyll, or it's just having impact on a chlorophyll, so the chlorophyll excited. So now we have a chlorophyll, excited chlorophyll like this. Okay, start. And when the electron is the we know the atom, for example, this is an atom X. When it excited to the X star, so that means the electron from a, for example, first cell. So one electron from first cell is entering into the second cell, or the if the energy is more, so it can jump to the third cell also. And if sometime there is a very high energy, so that time this electron can escape the uh, atom, and we call it a knockout electron. So we are not going to discuss about the knockout, but here from one cell to the second cell, when electron enters, so it will definitely come like this. Okay, so the first thing, it will directly come to the its own cell and emit the all energy which is having because of the absorption in a single stroke, and this energy is coming from the electron h nu. Okay, so guys, let's see, this is not a very efficient process to emit the all energy in a single stroke. But if you see, the electron is coming like this, so the third cell to the second one, and second one to the first cell. So at each and every jump, it can emit the energy. Okay, so like this, here is a 1 h now, and here is the second one. Okay, so the two time we have the energy, and that is what we call it a phosphorus. So the phosphorus, we this is called as a fluorescence. Okay, so the in a single stroke, the electron is going to its own shell. Okay, so the excited electron is going to its own shell in a single stroke, emission of a single stroke in energy. Okay, so if you see, the energy is emitted by step, so it is more I mean the more convenient for the photosynthesis, and that's why we required photo the sorry this is called as a fluorescence, and this is called as a phosphorus. So we required phosphorus. Okay, so let's say the what happened. So the photon is coming, a photon means the packet of light, you can say it is a packet of light. Okay, so the it is packet of light which is coming from the sun. Okay, so when you use the photon, the quanta word, so you are talking about the corpuscle theory of light. Okay, so you are not talking about the wave theory of light here. So guys, when it is coming, but you can consider it also. Okay, so it doesn't mean that it is not working with the photosynthesis. Yeah, it works. So the energy here is the excitation happen and the energy is transferred to the next one. So the next one, so it doesn't matter, it is going to like this. Sometime it can move like this also, so directly into uh, the reaction center. Okay, sometime it is a move like this, so from here to here, here to here, here to here. Okay, so again here to here, from here to here, and it may be come again to the first one, and it will escape the system and coming out from the leaf. Okay, so the escape. So it is also happen inside of the reaction center. So the reaction center, if you see this the excitation process or the continuation of excitation process is a random. Okay, so it do not have any direction. It doesn't mean that it is moving always moving toward the reaction center. No guys, if uh, uh, the probably the reaction center found in the path of excitation, so it will definitely absorb the electron, otherwise it is moving like this. Okay, so the photon is randomly migrate toward the reaction center and rarely they miss the center of a reaction, and they re-emitted inside of the environment. So this is called as a re-emission. Okay, so this is a re-emitted a photon. Okay, so the photons are coming from the light, so they are moving toward the reaction center. So some can find their reaction center, so for example here, here. But if you see this one do not find the reaction center, he miss first here, and then moving randomly toward the anywhere in the chlorophyll and finally escape the chlorophyll. Okay, so this is how the reaction center can be missed from this the pigments. Okay, so here is your the spectra of light. Okay, so it is electromagnetic spectrum. So the we are interested in this light only, so the visible region. Okay, so I am going to explore this visible region from violet to the red color. Okay, so this is only the region where the light is absorbed by the photosynthesis. Okay, so the photosynthetically active region, we call it a bar, photosynthetically active region. Okay, so the blue light is more and the red light is more necessary. So the blue and red light are more necessary for the absorption, sorry, for the photosynthesis process. Okay, next let's see next one. Photosynthesis consists of both light reaction and the dark reaction. Okay, so what is meant by light reaction and what is meant by dark reaction? So I already explained, but one more time we have the few text here. So the light reaction capture a light energy and convert it into the chemical energy in the form of reducing potential, that is the NADPH. Okay, so that actually we write it like NADPH plus H plus. This is the reduced form, and ATP with the evolution of oxygen. So this is your light reaction. लाईट रिएक्शन परत परत विचारायचं नाही, लाईट रिएक्शन काय करते, तर या दोन गोष्टी बनवते, NADPH plus H plus and ATP. So these are the product of a light reaction. Products of light reaction. Okay, so the dark reaction, if you see use NADPH, so this is using NADPH. Okay, and ATP to drive the endergonic process of hexose sugar formation from the carbon dioxide in a series of reaction in a stroma. So this is nothing but the dark reaction. Dark reaction, we called it a Kelvin cycle, Kelvin Benzene cycle म्हणतो आपण त्याला. Okay, so these two products are consuming by the dark reaction. So it is again converted into the NADP, and this is converted into the ADP. Okay, so the what is the output is the carbon dioxide is converted into the glucose. glucose from carbon dioxide and H2. So this is the actually process. We called it the photosynthesis. So it involved both light reaction and the dark reaction. The product of a light reaction is used as a source of energy by the dark reaction, and the glucose is synthesized inside of the plant cell. Okay. So the photosystems, there are two type of a system which is involved in the absorption of light, guys. Okay, so the PS1, we call it a photosystem 1, which is absorbing 700 nanometer of light. 700 nanometer. 700 nanometer of light means what? It is a lambda, guys. It's a wavelength. Okay, so a lambda is equal to, so isn't look like a lambda. So I'm going to write here PS1 700 nanometer. It's a lambda. And PS2 lambda is equal to 680 nanometer. Okay, so all chlorophyll is a part of either light harvesting complex or the PS1 or the PS2. Okay, so the chlorophylls what we study earlier, so these chlorophylls are nothing but the working as a one, that is the photosystem, working as a photosystem, or the light harvesting complex. Either they are involved in the absorption of light, or they are involved in the transport of electron. Okay, so they are either part of a light harvesting complex or the chlorophyll. Okay, so the PS1 absorbs 700 nanometer wavelength, that is a lambda, and 680 by the PS2. Okay, so the chloroplast given light at at 680 and the 700 nanometer simultaneously yield more oxygen than the sum of amount of when each is used alone. For example, this is a chloroplast, and you are giving the light to this chloroplast. Okay, so if you are giving only blue light, so the oxygen which is evolved from the chloroplast is a sign, or it is a product of the light reaction, and that's why we measure the oxygen. So the measuring of oxygen will which give us the information about the blue light. Okay, so about the photosynthesis. So if you expose the chlorophyll to the blue light, so it will result into the some amount of oxygen. If you expose this chloroplast to the red light, so it will again result into the making or the yield some amount of oxygen. Okay, but if you expose this chloroplast to the both light blue and red, म्हणजे for example तुम्ही chloroplast ब्ल्यू लाईटला एक्सपोज केला तर थोड्या अमाऊंट मध्ये ऑक्सिजन भेटणार आहे, रेडला एक्सपोज केला तरी थोड्या अमाऊंट मध्ये ऑक्सिजन भेटणार आहे. But if you expose this chloroplast to the both light, the blue and red at a time, so it will definitely give you the more amount of oxygen, and that's why the efficiency, if you see of a photosystem, it is depending upon the quality of the light and the intensity of the light. So light should have the both region inside, the blue and the red. Okay, so it is very necessary for the synthesis of sugar. Okay, so let's see water is a donor uh, for the electron. Okay, so the PS1 and PS2 they required electron, otherwise the electron transport chain will do not have any electron to transport. So there we need the I mean donor. So your water is act as a electron donor. Okay, so S2O is finally split into the, for example, 2S2O finally split into the H plus and the oxygen here. Okay, so you can the balance the reaction as per your knowledge of chemistry. Okay, so I am not going to balance that reaction here, but I have the few steps to shock you like how the chemistry is working here inside of the biology. Okay, for photosynthetic NADP plus reduction, the photosynthetic bacteria use H2S. Okay, so we know that we sometimes call it the chemosynthetic bacteria. Isopropyl or the other oxidizable substrate, so they use this the S2S or the other substrate like this isopropanol, they use as an electron donor. Okay, but the oxygen we depends upon oxygen we depends upon is in a turn on large amount of photosynthesis on earth. Okay, so this oxygen we depends upon it, so it is coming from the photosynthesis. It is always product of a photosynthesis. Okay, so let's see oxygen evolution by ps2. So now the guys the what we call it the um, here is a twist. Okay, so the ps2, that means the photosystem 1 is not going to work first, ps2 is going to work first. Okay, so why, I am going to explain you later. So ps2, it is the p68. We know that it is absorbing the light of 680 nanometer, so we call it p68. Okay, uh, sorry, p680. So we also call it a p680. It is a cycle through the 5 oxidation state. Okay, so the oxygen evolution means the conversion of a H2O to the O2. Okay, is a cycle of a 5 oxidation state. 5 oxidation states. Okay, so the one electron is removed in a state. So the for first 4 step, one electron is removed, so the 4 electron are coming from the water. Okay, so the single molecule of water giving you the 4 electron in a 4 oxidation state, and this 4 electron required for oxidation state. Fifth step involve the H2O oxidized to the 4H plus and the O2. Okay, so the finally we will have the H2O water, which is converted into the 1 oxygen and 4EH plus and the 4 electron, guys. Okay, so let's see what happened in the next stage. So this electron which is coming from the water is now we have 3 in our hand. Okay, so here we have the 4H plus and the electrons also. So this electron is entering into the p680. So this is your ps2. Okay, so I am also written here ps2, so which is absorbing the light of uh, okay, so this is a light of having the 680 nanometer lambda. Okay, this electron is excited here, so this is the energy actually h nu. You know that what that h nu means, and this is a star, so the star is actually the sign of excitation. Okay, so this ps2, okay, which is working first. So why which is working first, guys, when the scientists discovered the photosystem, so the photosystem 1 is discovered first because the scientists found that the photosystem 1 having the absorption power of light at 700 nanometer, and he explained it very beautifully. But later we found that the ps one more ps is already working before the ps1, and which is actually giving or the act as an electron donor for the ps1, and that's why the ps1 is a name given to the second because it is found first. Okay, and the ps2 it is found later, so we call it a ps2. But if you see the working mechanism, the ps1 is a first in a working. Okay, it is a first, and ps1 is a second. Okay, it is coming coming at the second position. Okay, so the ps1 and sorry ps2 is now excited here, and this excited ps2 donated its electron to the chlorophyll a. Okay, so this is a chlorophyll a, and then chlorophyll a donated its electron to the pheophytin a. Okay, so pheophytin a to quinone a, quinone a to quinone b, and it is finally entering into the cytochrome B6F or the cytochrome BF complex. Okay, so this is the two molecules actually. This is cytochrome BF complex. So this cytochrome BF is also called as the pump. Okay, so it also called as the pump. It is nothing but the pump, guys. So this is involved in the pumping of H plus. So H plus is coming inside of a thylakoid from the stroma, and the 700 at the PC. This is a plastocyanin. Okay, so the plastocyanin is uh, the molecule which is having electron, or the accepting electron from the cytochrome B6F, and donate to the P700, and now the P700 having its light for the absorption, that is the 700 nanometer. Okay, and then it emit, or it excited toward the P700, and then the next electron transport chains comes into the focus, and it start the donation of electron to the last one, and we have pyridoxine molecule at the final, and then pyridoxine to the uh, pyridoxine reduction molecule. Okay, so the here NADP plus 2H plus comes together, and they form the compound like a 2NADPH plus H plus. Okay, so this is the reducing power uh, which can be used to synthesize the sugar from the carbon dioxide, and that's why we draw here H2O, because H2O is already used here. Okay, so guys, this is the actually complete process of photosynthesis, and this is the cyclic process. So if you say, it is looks like a jade. Okay, it is looks like a jade, and that's why we call it a jade scheme. Sometime it is called as a jade scheme. So it is like this, and that's why we called it a jade scheme. Okay, यामुळे याला jade scheme म्हणतो आपण. हा. आणि दुसरी गोष्ट, जर तुमचा फेरेडॉक्साईन इथून डिरेक्ट इलेक्ट्रॉन बाहेर न देता वापस जर परत याला देत असेल इथे, ओके, क्यू पूलला, या क्यू पूल मध्ये, तर सायक्लिक होतं. सायक्लिक मध्ये फक्त एटीपी बनतात. ओके, सो देर आर टू टाइप ऑफ the electron transport chains. Okay, so the first one is a non-cyclic. Non-cyclic gives you the two things, NADPH plus H plus, and the cyclic one give you the two things, sorry, one only ATP. Okay, so there is no any NADH, NADPH plus H plus is not going to be synthesized into the cyclic process. Okay, cyclic process is only having the synthesis of ATP. When ATP is NADPH is not required for the plant, that time the cyclic process is a very efficient to synthesize the ATP. Okay, so the cyclic process is happening inside of a PS1 only. So the cyclic photosynthesis, or the cyclic the electron transport system, is the property of a PS1. Okay, so please keep in mind that it is a property of PS1. Okay, सायक्लिक प्रोसेस फक्त PS1 मध्ये होते, लक्षात ठेवायचं सगळ्यांनी. PS2 चं नाव काढायचं नाही सायक्लिक प्रोसेसला, नॉन सायक्लिकला PS2 तुमची आहे. ठीक आहे, ऍक्च्युअली हा टोटल ही डायग्राम खूप डिटेल आहे. ही डायग्राम जर तुमच्याकडे राहिली तर माझ्यामध्ये तुम्हाला खूप जास्त फायदा होईल याचा. चलो नेक्स्ट, परत तीच डायग्राम तुम्हाला या ठिकाणी दाखवलेली आहे. त्याच्यामध्ये काय थोडा सुटसुटीतपणा आहे, मोकळ्या सगळ्या गोष्टी आहेत ज्या करून तुम्हाला लक्षात ठेवायला सोपं जाईल. हा पंप आहे, लक्षात ठेवायचा, विच पंप्स दी H प्लस. सायटोक्रोम B6F जो आहे ना, हा पंप आहे H प्लस, याला आपण प्रोटॉन पंप म्हणतो. ठीक आहे, सायक्लिक प्रोसेस इथं दाखवलेली आहे, ही सायक्लिक आहे. हा जो एरो आहे हा सायक्लिक आहे. ठीक आहे, आणि इथे फेरिडॉक्सिन रिड्युसिंग कॉम्प्लेक्स आहे, FD स्टँड्स फॉर फेरिडॉक्सिन. बाकी हे सगळे इलेक्ट्रॉन ट्रान्सपोर्टर आहेत, त्यांची सगळ्यांची नावं तुम्ही याच्या आधी पण पाहिलेली आणि आपल्या लास्ट लेक्चर मध्ये पण आहेत, आणि आपण परत एकदा तुमच्या इलेक्ट्रॉन ट्रान्सपोर्ट सिस्टम मध्ये ज्यावेळेस जाऊ आपण मायटोकॉन्ड्रियाच्या, तर परत एकदा रिवाइज करणार की नेमकं काय होतं. ओके, हा जो 1 वाला आहे हा एक्सायटेशन आहे, इथे इथे पण इलेक्ट्रॉन एक्सायटेशन आहे. इथे 600 ची लाईट ऍब्झॉर्ब केलेली आहे, इथे 700 नॅनोमीटर ची लाईट ऍब्झॉर्ब केलेली आहे. ही PS2 आहे and he PS1 आहे. So the cyclic photophosphorylation ATP without NADPH. Okay, so the photo excited electron removed from P700 returns to the P700 in a pathway indicated by the dashed line is your cyclic process. Okay, so the cyclic photophosphorylation depends only on a PS1 and not a PS2. Okay, so it is a property, cyclic is a property of a PS1. It is completely depending on PS1 and not PS2. Okay, so guys, this was the light reaction which is turned into the ATP and the NADPH. It is a reducing power and it is a high energy compound, that is currency. Okay, लक्षात ठेवायचं ATP आणि NADH, synthesis of ATP and NADH, the ultimate aim of your light reaction. Okay, so guys, thank you very much. So see you today at 7 p.m. evening. Okay, so we will have the mid and the hindbrain in today's lecture, so we will discuss it there. Yes, so thank you very much and have a good time. Bye bye.